Hello everyone, welcome to Pat in the Hat. I'm Pat, and today we're talking about GC content, or in layman's terms, just counting the number of G's and C's in a sequence. So the idea for this was to create a tool for myself to basically use a FASTA file as a input for a program and for it to just spit out the GC content of the sequences in that FASTA file. So for those of you who aren't familiar with FASTA files, it's a very standard file format where there is just a number of sequences here and then basically a sequence ID. So in this case, I've downloaded this FASTA file that contains the FTO gene. And this happens to be for cows. So this is the test my test file that I'm going to use for this project. So let's get started. I'm going to be coding in Python, and so let me open my text editor here. I use Genie. Genie is the closest thing I can find to Notepad++ on Linux, and it seems to work pretty well. So I'm going to go ahead and save this file as just in my desktop here as gccontent.py. Okay. So now we're good to go. So the first thing I want to do is I want to use the FASTA file as an argv argument. So I can just put the name of the file after my program and execute, and it'll do it all for me. So to do that, I'm going to have to import sys. This is a very standard package. Most installs of Python should have this pre-installed. If you don't, it's pretty easy to install. Next, I need to open the file. So what I'm going to do is say FASTA file is equal to open, and then I'm going to do sys argv element1, and we're going to read that file. So all this is doing is it's taking the first argument, which is going to be our file name, and it's going to go ahead and open that file. A few things that I'm going to need for this, since I'm going to be counting the number of G's and C's and A's and T's, I need to set some variables here. So I'm going to set GC equal to 0. I'm going to set AT equal to 0. And then I'm going to set unknown equal to 0 as well. So unknown, sometimes you have a nucleotide that is not known in a FASTA file, and it's usually designated by an N. So that's what this category is going to be for. So I'm basically going to be counting the number of G's and C's, and I'm going to be adding, for every G or C that it finds, I'm going to be adding 1 to this variable. For every A and T that it finds, I'll be adding 1 to here. For any N that it finds, I'll be adding 1 to this variable. So we have some if statements coming up for those of you who are familiar with Python. So the first thing I need to do is I need to open our file. So I'm going to say for line in FASTA file. So now we're going to iterate through the FASTA file. One tricky thing here, let me open this back up. How I want this program to work is I want it to print out this ID line, and then I want to print out the percentage of GC. So one thing that I need to do is I need to, in order to print out this line, I'll need to save this line for later, because the first line is this ID line, and I don't know the GC content of this yet. So I'll need to save this, find the GC content, and then actually print it out when it hits this line here. So it'll actually, getting to this line, I'll print out all the information for this. When I get here, I'll print out the information for this sequence here. So the first thing I want to do is actually an if statement. So if line starts with then I'm going to and I want to do this I want to skip the first line, so I'm actually going to say GC plus AT if it's greater than 0. So in that first iteration, the first line that we pick up, this is not going to come true here. But this is where I want to actually do most of my calculations. So I'm going to do GC plus AT. That's going to be our total. So the total number of nucleotides that we find. And then our percentage is going to be So here what we're going to do is, so we haven't got to the iteration yet. This is all going to happen. This is all revolving around printing out what we want it to print out. So when it hits these lines, that's when this is going to run, except for that first line. Because at the first line, we don't have any information yet. And then I'm going to print our, I'm going to call it seq ID, and then percentage. So we don't have a seq ID variable yet, but that's because I'm going to declare it right here. So seq ID is going to be equal to just line, and I'm going to take away, use the strip function to take away that new line character. So if you imagine we're going through our file here, and for that first line, it's going to get here. Yep, it starts with this, so it'll go down, but it will not execute this stuff. All that first line is going to do is just save that ID into seq ID. And then another thing I want to do is I need to, when this happens, I need to reset the counts. So reset counts, and I'm just going to take this, put these here. So I need to reset these. So the GCs aren't compounding over multiple sequences. So each sequence is treated as an individual unit, and we're not getting the GC content for all the sequences combined. Okay, so that's all. This all happens just on the sequence ID. Now let's look at the other line. So if it does not start with the arrow character, we're going to do else. And then I'm going to call it nucleotide string, not the most original, but whatever. And I'm going to make a list out of that line. So what this does is it just turns this, let me open that back up. So it turns this into a list. So each one of these letters or nucleotides is going to be an element of that list. So that's nice because we can now iterate through that list. So now I'm going to do another for loop, for n for nucleotide, in nuke string. And then here comes our lengthy if statements. So I'm going to say if n is equal to g, or n is equal to, I'm also going to put lowercase g, because FASTA files can have uppercase and lowercase, and the reason for that I'm not going to go into in this video, but just trust me. N equals to C, or N equal to little c. Okay, so if it's equal to any of these, then what we're going to do is just do a simple GC add 1. Okay? If n is equal to capital A, or n equal to lowercase a, t, or n equal to lowercase t, then we're going to do our at plus or equals 1. So all this is doing is it's looking through that list, and it'll take the first element, it'll see if it's an A, C, T, or G, and whichever it finds is going to add to that variable that we created at the beginning. And then finally, else, we're going to have it add to our unknown. And remember, these are usually represented by an n in the FASTA file, so if you wanted to, you could say another else if statement and say else if n is equal to n, then unknown plus 1. But in this case, we're just going to use a simple else statement, kind of as a catch-all. So that should be the majority of the program here. So for those ID lines, it's going to go ahead and print out, do the calculations that we want it to do, get that percentage of GC here, and then print that out. And one thing we do need to do is something I had thought about is there's no ID line here at the end of the statement, so it's going to be calculating the GC for this, but then we don't have another ID line at the end where it's going to get printed out. So I actually need to repeat a little bit of my code here. So what I'm going to do is I'm just going to say this calculates for last sequencing file. Okay, so our total, I'm just going to actually, maybe I can just copy and paste this, because I just need to take these 3 lines. So for that last, once the file has finished, we've gotten through the whole thing, it'll go ahead and take our GC and AT and do those calculations one last time and print out one last time. So this should be our finished product here. So let's take a look and see how it works. So let me save this. I'm going to go to our terminal here. So let's see what we have. We just have our FASTA file here, and then our program that we wrote here. So all I'm going to do is just say Python GC content, and then use this again as, we're just going to use this as an argument. So FTO, and then I should be able to hit enter, and it'll print out those. Oops, I do have a syntax error here. So I always do. Let's see what it is. So line 14, percentage seek ID. Let's see. I think that should be okay. Oh, okay, I'm just missing no parentheses here. Got it. Usually what it is, something small. Okay, let's save that and give it another try. Oh, one more. Okay. Probably the same thing. Oh yeah, I copied and paste, so it's gonna be the same thing. Okay, so here it is. So we get our ID line, and then right after we get our percentage. So in this case, we see the sequence at 46% GC, 53, 43, 37, 52, and 46. And that's it, guys. Let me know if you have any questions in the comments, and I'd be happy to take recommendations for future videos. Thanks, guys.